Student, today topic is antiviral drugs. Uh, s- so this is also very important topic for your examinations. In antiviral topic, you need to discuss the three drugs. So one is majorly anti-HIV drugs, antiretroviral drugs. Second one is anti-influenza drugs. Third one is anti-herpes drugs. Okay, so today in this lecture we are going to be discuss three main topics: antiretroviral, so anti-influenza and anti-herpes drugs. Okay, so first is antiretroviral drugs. Main AIDS. Okay, so antiretroviral drugs mainly used for the treatment of AIDS. Okay, acquired immunodeficiency syndrome, all of you know that. So AIDS is caused by virus, that is virus is called as HIV virus. Okay, AIDS is acquired immunodeficiency virus, is caused by HIV. HIV stands for human immunodeficiency virus. Okay, so why is going to be suppress the immunity? Because it mainly affect the WBC cells. Okay, WBC cells, it mainly affect the WBC, especially it contain CD4 receptors. Okay, CD4 receptor positive cells. Okay, CD4 receptor positive cells, CD4 receptor positive cells. Okay, so in this diagram, so upper one, so this is majority of the what are the targets of the target cells of HIV is any cell that contain express CD4 receptors. Okay, together with the co-receptors. Okay, chemokine receptor, also called as a chemokine receptors. So these principal targets of HIV virus are CD4 containing T lymphocytes, monocytes, macrophages, and cytotoxic T lymphocytes. Okay, so with this is going to be will suppress the immunity. Okay, so what are the characteristic of HIV is immunosuppress is the major characteristic. Because of immunosuppression, there is a chances of getting so many opportunistic infections. Next, there is the development of neoplasm, secondary neoplasm cancers. Third one is neurological manifestation. These three are the major characteristics of HIV. Okay, so this HIV, so this is the life cycle of HIV. Like uh, so I will discuss some structure of HIV particle. So HIV, it is consist of two single stranded RNA genome. Okay, two single stranded RNA. So along with the RNA, it contains three essential enzymes such as reverse transcriptase, integrase, protease enzyme. Okay, it contains nucleic acid in the center. Okay, along with nucleic acids, which nucleic acid it is a retrovirus, it is a RNA virus. Okay, RNA containing virus. Okay, it contains of two single stranded RNA along with the three essential enzymes like reverse transcriptase, integrase, protease enzyme. Okay, this nucleic acid is surrounded by one layer that is called as a nucleocapsid. Okay, again this layer is covered by one more layer that is called as a matrix protein. Later on lipid membrane. So this lipid membrane is studded with, and it is studded with surface glycoprotein. So this surface glycoprotein, okay, this is surface glycoprotein. The glycoprotein 120, glycoprotein 41, these two combined together called as a glycoprotein 160. Okay, the outer membrane of the virus, it is studded with surface protein, that name is called as glycoprotein 160, is divided into one GP 120, glycoprotein 120, glycoprotein 41. Okay, so these glycoprotein generally useful for binding with main CD4 receptors and co-receptors. Okay, here I am going to discuss about the life cycle of the HIV. First is, what is the first step? Here the HIV virus is going to be bind with, okay, CD4 receptors containing cells okay, with help of glycoprotein 120. Okay, so later on, so it is undergo infusion. Okay, so first binding, later on infusion means penetration. Okay, penetration. So after penetration, there is a penetration. After penetration, there is a release of single standard RNA. That single standard RNA, the uncoating is nothing but removal of all the codes. Already I told na. So one nucleocapsid is there, one matrix protein is there. Okay, so outer membrane is there. All the codes will be automatically dissolved. Okay, when it is incorporated into the T lymphocytes. Later on, there is a release of, okay, there is a release of a single standard RNA into, okay, cytoplasm of our T lymphocytes. So where this RNA converted into DNA. Okay, where this RNA converted into double standard DNA in the presence of virus specific enzyme called as a RT. Okay, what is RT? RT is nothing but reverse transcriptase. Okay, so so what reverse transcriptase do? It will synthesize DNA from RNA. Generally, RNA is synthesized from the DNA. Okay, but here, okay, the DNA is synthesized from RNA. So this is the reverse of the transcription. So that is called as reverse transcription. Okay, whatever the enzyme involved in the reverse transcription is called as a reverse transcriptase. Is nothing but DNA polymerase. Is also called as RNA dependent DNA polymerase. Reverse transcriptase के इंको पेर ఏంటంటే RNA dependent DNA polymerase. Okay, it is a DNA. Why DNA polymerase you are stressing? Because it synthesizes the DNA. That's why it's called as a, okay, RNA dependent DNA polymerase. Okay, so this is the first use of the reverse transcriptase. Okay, next, after synthesized DNA, the DNA is going to be, okay, it is a fusion with our host DNA. Okay, whatever the viral DNA is produced, the viral DNA is going to be, okay, incorporated into the host DNA. T lymphocytes low, whatever the DNA present in the our host T lymphocytes, the viral DNA is going to be incorporated. Okay, so later on, whenever the DNA undergo transcription, okay, so viral DNA undergo transcription and produce both viral genome RNA, genomic RNA, and as well as the viral specific mRNA. Okay, so after during transcription of this viral DNA produce two product. What one product is, so the genome of the RNA, okay, genome of the new virus RNA. So later on, so viral mRNA. Okay, this viral mRNA in the presence of our ribosomes produce some protein. Okay, that is called as a GAG-PAL protein. So these proteins and as well as the viral genomic RNA, they are assembled at one place. Okay, very near to the cell membrane. So these viral RNA and as well as the GAG-PAL protein, they are going to be released from the infected T lymphocytes in the process of budding. Okay, so so that they leave the virus from the host cell through process of budding. Okay, once it is released from the infected cells budding, so it is not going to be infect to the other T healthy lymphocytes. Okay, so in the process, the whatever the virus immediately released from the T lymphocytes immediately after replication is called as a immature viron. Okay, immature. Why immature viron means it does not contain any structural component. Okay, it is not going to be contain any structural component available to attach to the host cell, so that it converts from, okay, immature viron to mature viron in the presence of, okay, protease enzyme. Okay, so already we know the use of reverse transcriptase. Okay, so what is the role of integrase the viral DNA incorporated into host DNA. So the only enzyme remaining in the virus specific enzyme is protease. The protease will convert immature viron to mature viron. Okay, immature viron to mature viron. So what protease do? Okay, protease, already I discussed during the replication process, viral genome is produced, as well as the, okay, some one protein is produced, very big protein. That protein is called as a Gagpal protein. That protein is going to be break down into various structural and functional proteins. Okay, that Gagpal protein is cleaved by protease enzyme into structural proteins. What are the structural proteins of the virus? Already we discussed starting of the discussion, like glycoprotein, okay, nucleocapsid, matrix protein. These are all the structural proteins. Okay, so these are responsible for the making of virus. Okay. Yes. What are functional proteins? Functional proteins means whatever the proteins which are involved in the some reactions. Okay, so for working of the virus, okay, is called as reverse transcriptase, protease, integrase. Okay, so after this process happen, after cleaving of, okay, that Gagpal protein, a big protein, is cleaved, okay, or cleaved means breakdown into, okay, various structural and functional proteins in the in the presence of virus specific protease enzyme. So when it is happening, the virus will be converted from immature viran to mature viran. So what is the significance of maturation process is, if the particle immediately after releasing from the infected cell is unable to infect the other healthy T lymphocytes, okay, so that it is an uninfected one. Okay, so so maturation process is the step where which converts, okay, the virus particle converted from uninfected to, okay, so it is called as a mature viran. This mature viran is going to be capable of infecting, okay, other T lymphocytes. Okay, again it is binding to other healthy T lymphocytes. Again the life cycle is so on continues. Okay, this is about the life cycle of HIV. So if you understand the life cycle of HIV, it is easy to understand the so antiviral drugs. Okay, so later on, whatever the classification of drugs, below the diagram, please carefully watch. So first, now what are the areas you are going to be as a targets, okay, to stop the replication of the DNA. Okay, so first step is entry inhibitors. These are the drugs will stop the entry of virus into, okay, entry of virus into the T lymphocytes. Okay, they will stop the entry of, so that example, okay, N-furatide and Maraviroc. So these are going to be impair the process of binding and integration. Okay, binding and integration of virus. Okay, later next is reverse transcriptase. This is the enzyme where which converts, okay, viral RNA into viral DNA. So this is the process, this is the process occur before replication of the virus. Okay, so that in case if you stop this area, okay, if you stop the further process like integration, transcription, okay, budding, okay, so releasing and infecting, so on, so on. So this is called as a reverse transcriptase enzyme inhibitors. So our target is here is enzyme. What is the enzyme name is reverse transcriptase. Okay, so this reverse transcript enzyme inhibitors, again three types. One is nucleotide reverse transcriptase enzyme inhibitors, nucleoside reverse transcriptase enzyme inhibitors, non-nucleoside reverse transcriptase enzyme inhibitors. Okay, please remember nucleotide and nucleoside. Okay, so what is the difference between the nucleoside and nucleotide? So this nucleoside is nothing but they have uh, nitrogen base and as well as the phosphate backbone. Okay, they don't have sugar. Nucleotide means they contain three nitrogen base. So actually reverse transcriptase enzyme inhibitors are nucleotide and nucleosides are nothing but they are anti-metabolites. So these are actually prodrugs. Actually they are prodrugs. So they are looking like our natural metabolite present in our cell. Already we discussed about anti-metabolite topic in case of anti-cancer drug. So the same mechanism is going to be executed by this class of drugs. Okay, nucleotides, reverse transcriptase enzyme inhibitors, nucleotide. Okay, tenofovir, only one drug. Nucleosides, so many drugs. So these are more popular drugs, nucleosides. Okay, so abacavir, didanosine, emtricitabine, lamivudine, stavudine, judovidine and all. Okay, Next is non-nucleosides. Okay, nucleotide, nucleoside, non-nucleoside. Example: dilavirin, efavirenz, nevirapine, and etravirin. Okay. So next class of drugs are integrase inhibitors. These are the drugs. These are working on. Uh, these are working to integrate the viral DNA into host DNA. So example, only one drug is called as a raltegravir. Next, protease inhibitors. Already we discussed protease. So it will stop them conversion of immature to mature viral. Okay, these are also very important. Okay, nucleoside reverse transcript enzyme inhibitors, protease inhibitors are okay, very important. So protease inhibitors example: indinavir, lopinavir, ritonavir, saquinavir, nelfinavir, tipronavir. Okay, these are the drugs. Okay, classification of drugs which are used for the treatment of HIV. So among these, so please more concentrate on reverse transcript enzyme inhibitors and protease inhibitors for exam point of view. Okay, so now we are going to be talk on okay uh, individual anti-HIV drugs. Okay, so individual anti-HIV drugs, so reverse transcriptase enzyme inhibitors. Okay, see, so are able to see that topic. So nucleoside reverse transcriptase enzyme inhibitors. Okay, reverse nucleoside transcriptase enzyme inhibitors. Okay, so what is meant by this nucleoside reverse all nucleoside reverse transcriptase are have the same mechanism. Okay, so same mechanism of action. So different drugs in the class serves as anti-metabolites of different and purine and pyrimidine bases of the DNA. So these are the anti-metabolites. Okay, so these are the anti-metabolites. Anti-metabolites. These are the anti-metabolites of purine and pyrimidine nucleotides. Okay, so these are synthetic nucleoside reverse transcript enzyme inhibitors are synthetic derivatives of naturally unlocks of nucleosides. So this is okay our drugs. Okay, whatever the drugs we are called as a nucleoside reverse transcript enzyme inhibitors are synthetic derivatives. Okay, synthetic derivatives, they are look like they are analog of naturally occurring nucleosides. Okay, so our drugs are look like a naturally occurring nucleoside, so that they compete with, okay, our drugs are compete with naturally occurring nucleosides. Okay, in the process of competition they are going to be bind with, okay, they are going to be bind with reverse transcriptase. Okay, so what is reverse transcriptase? Is a DNA polymerase. It synthesizes the DNA from RNA. It synthesizes the viral DNA from viral RNA. So in the process our drugs bind with reverse transcriptase. Okay, so after that the reverse transcriptase enzyme inhibited, so there is no production of viral DNA, so replication of virus is going to be stopped. Okay, so that you have to so that the virus if infection will be reduced. Okay, so this is the examples. Next is mechanism. Okay, mechanism how nucleoside reverse transcript enzyme inhibitors do at working. So all nucleoside reverse transcript enzyme inhibitors are prodrugs. Okay, prodrugs. Okay, these are converted, these are nucleosides means they contain nitrogen base, uh, sugar. Okay, sugar. Okay, so that they need to be undergo some phosphorylation step. Okay, so all nucleoside reverse transcript inhibitors, they are undergo phosphorylation step by host cell kinases, our kinases. Okay, these what kinases do, they convert nucleoside to nucleotide. Okay, nucleoside to nucleotide. Okay, once after addition of phosphate. Okay, so now it's called as a the product is called as nucleotide. Okay, so all nucleoside reverse transcript enzyme inhibitors undergo phosphorylated by host cell kinases to convert it into active triphosphate form. Okay, for example I will take one drug here. Okay, suppose judovodin. Okay, judovodin in the presence of host cell kinase first is converted into judovodin monophosphate. Okay, later on judovodin diphosphate, later on judovodin triphosphate. Okay, judovodin triphosphate is the active. Okay, judovodin triphosphate is the active form of the judovodin. Okay, so this it is compete with, okay, it is compete with corresponding nucleoside, endogenous nucleosides for the active sites present on the active site present on the reverse transcriptase, so that either it inhibits the reverse transcriptase or either is incorporated into the viral DNA. So later on the viral DNA is going to be non-functional. Okay, so this is about the mechanism of action of all nucleoside reverse transcriptase enzyme inhibitors. So the problem is, so this nucleoside reverse transcriptase inhibitors not only inhibit the viral DNA polymerase and also inhibit the our host cell DNA polymerase enzyme, so which leads to some side effects. Okay, DNA polymerase present in the mitochondria, it appears to be it appears to be causes some side effects in the host cell. Okay, next, pharmacokinetics. Okay, so pharmacokinetics all are given by orally. Zidovudine is also given by intravenously. So they are mainly eliminated primarily through renal excretion. Okay, they undergo metabolism. Metabolism. Okay, see, this is okay. This is the continuation. Continuation of the paper. Okay, so this metabolism generally zidovudine abacavir metabolism by glucuronational transferase enzyme. Okay, zidovudine and abacavir, they are metabolism by glucuronide transferase enzyme. Okay, so why I am tracing here metabolism? So there is this drug interaction happen with other drugs. That's why I trace the metabolism of the zidovudine. Okay, adverse effects. Okay, so majority of the adverse effects is due to inhibition of mitochondrial DNA polymerase in the certain tissue. So very important toxicities are, so one is peripheral neuropathy. Peripheral neuropathy. So pancreatitis, lipodystrophy, liver toxicity, lactic acidosis, hepatomegaly. So these are the major toxicities, specific toxicities towards nucleosides. Again, I will repeat. So peripheral neuropathy, pancreatitis, lipodystrophy, liver toxicity, lactic acidosis, hepatomegaly. So these are the other side effects. Okay, principal common unwanted side effects. Okay, it is not particular to one drug and two drug. This is the side effects which are common for nucleosides. GI related problems, CNS. Okay, already neuropathy we discussed. Okay, dermatological means just simply rashes, urticaria. Musculoskeletal means there is a myalgia, okay, arthralgia, joint pain and muscle pain. Blood disorder, definitely. So in any examination, any there, if there any side effects of blood disorder, majority they decrease in the RBC, WBC platelets, anemia, thrombocytopenia, and this metabolic already you discuss, lipodystrophy, lactic acidosis, pancreatitis, and liver damage. Okay, so this is about means adverse effects of nucleoside drug interactions. So drug interactions mainly occur because zidovudine mainly undergo glucuronation in the liver, then eliminated through urine. Okay, here zidovudine toxicity, AZT means zidovudine. Okay, AZT means zidovudine. Okay, zidovudine toxicity is going to be potentiated by other drugs which undergo same glucuronation metabolism. Example, probenecid, paracetamol, acetaminophen means paracetamol, lorazepam, indomethacin, cimetidine. Okay, suppose if you take zidovudine plus paracetamol, paracetamol will stop the metabolism of zidovudine. So it increases the plasma concentration of the zidovudine, cause toxicity, zidovudine toxicity. This is about nucleoside reverse transcriptase enzyme inhibitors. Okay, next coming to non-nucleosides. Okay, nucleoside and non-nucleoside. Okay, non-nucleosides, what is the major difference between the nucleoside and non-nucleoside means? So this non-nucleoside reverse transcript enzyme inhibitors does not require any phosphorylation step. Okay, they won't undergo any phosphorylation step, cellular activation. But nucleoside reverse transcript inhibitor, they require cellular activation by phosphorylation. Okay, initial phosphorylated into monophosphate form, second phosphorylation to diphosphate form, third phosphorylation to triphosphate form. Triphosphate is nothing but nucleotide of the reverse nucleosides. Nucleoside reverse inhibitors is going to be inhibit the DNA polymerase. But here is non-nucleoside reverse transcript enzyme inhibitors are, okay, they are highly selective, non-competitive. Nucleoside means they are compete. Why they are compete? Because they are structurally similar to that of natural nucleosides. Okay, so this non-nucleoside reverse transcript enzyme inhibitors highly selective non-competitive inhibitors of, okay, non-competitive directly it inhibit the, okay, HIV reverse transcriptase enzyme. Okay, they bind to reverse transcriptase site adjacent to the active site, so inducing conformational changes that results into enzyme inhibition. Okay, so please remember, non-nucleoside reverse transcriptase enzyme inhibitors, they bind to the reverse transcriptase enzyme, so other than the other than the active site, means it is binding to adjacent side to the active side. Okay, so that's why it is not a not a competitive, so it is an uncompetitive reverse transcriptase. Okay, it's bind to the adjacent side to the active site on the reverse transcriptase, which causes some, induce some conformational changes in the enzyme. Okay, so which results into enzyme inhibition. Okay, major, what is the characteristic? They do not require any activation by cellular enzymes. Okay, so kinetics, they are given by oral and rapid, good oral bioavailability, they are highly lipophilic, they reach to CNS also. Okay, so they are undergo metabolism and renal excretions. So major side effects, specifically non-nucleoside, is skin rashes. Okay, skin rashes, dermatological side effect, are the major side effects of non-nucleoside reverse transcriptase inhibitors. So these are example. Okay, please remember, these are the inducer, substrate and inhibitors. Okay, so here is a nevirapine and efavirenz is CYP450 enzyme inducer. Okay, dilaviridine is a enzyme inhibitors. Okay, means they are undergo metabolism in the liver, so they affect the liver enzyme functions. Okay, metabolic enzyme, nevirapine and efavirenz, they are enzyme inducers, and dilaviridine, they are in CYP450 enzyme inhibitors. So this is about reverse transcriptase enzyme inhibitors. Okay, we discussed nucleoside and non-nucleoside. Okay, so only one nucleotide is also there, it is also not require any activation, already it is a nucleotide form, so directly it is bind to the active site present on the reverse transcriptase, which causes enzyme inhibitions. Okay.